Hello, micro class. I'm Maddie, and with me we have Mia, Grace, and Joe, and we will be presenting new insights into the gut microbiome and loggerhead sea turtles, Caretta, stranded on the Mediterranean coast. A little background information: the Caretta is the most common sea turtle on the coast. This species of turtle is threatened by human activities that leaves thousands of them dead and many stranded on the coast. From another source, it's estimated that 44,000 die every year. Stranded and injured turtles need rehabilitation centers for life-threatening conditions. We will define stranded turtles from NOAA, that is, any turtle found on land or in water that is dead, injured, or exhibits any indication of ill health or abnormal behavior. The turtles will stay in these rehabilitation centers until they make a full recovery or die. However, from these recovery centers, wild turtles compared to stranded ones and ones in recovery centers have been found to differ in their gut microbiomes. And here we have just a map just to get a perspective of how many turtles are stranded on the coast of Sicily. And the larger the dot is how many more turtles were found in that area. The objectives of this study was to expand the knowledge of the gut microbiome of the loggerhead sea turtle, Caretta, and differentiate between wild, stranded, and rehabilitated turtles. The researchers wanted to characterize the gut microbiome of 9 sea turtles that were stranded on the Sicilian coast. They aimed to do this through high throughput sequencing analysis by targeting the V3, V4 regions of the bacterial 16S rRNA gene in fecal samples. And they targeted this region as it is where there's the maximum nucleotide heterogeneity in order to best characterize the microbial communities that were present. To explore the presence of the bacterial communities found in the loggerhead sea turtles, they then combined their results with other fecal data published from sea turtles stranded on the Tahirian coast in Tuscany and Liguria regions to obtain more of a complete data set. And these will be presented in our most important figure.
Some of the goals of the study included they wanted to determine the significance of the gut microbiome diversity in loggerhead sea turtles that have been stranded. They also wanted to determine the necessary components that make up this gut microbiome community and use this knowledge of loggerhead sea turtles in order to successfully rehabilitate those that wash up on shore. In order to do this, they collected stool samples from loggerhead sea turtles found along the coast of Sicily and processed within the first few days of the turtles arriving at the rehabilitation center. And then they extracted the DNA from the samples, and the researchers sequenced the V3, V4 regions of the 16S rDNA using primers associated with Illumina sequencing. DNA sequencing was used to determine the identity of the microbes present in the sample, and DNA purity and quality was assessed via spectrophotometry.
So for our presentation, we chose figure 4, and this figure is representing the different microbial components found in the gut microbiome of C. Caretta and comparing it to other studies taken in other parts of the world. On the y-axis is the percentage mean of microbial abundance, and on the x-axis is the different phylum of bacteria. The different bars represent different studies, blue being this study, red being one taken in the Tyrrhenian coast, gray being taken the Adriatic coast, yellow and light blue being taken the Australian coast. As we can see, the levels of Firmicutes found in the turtles in the blue, red, and gray studies have significantly higher concentrations than those taken in the yellow and light blue studies. Proteobacteria values in the study and in the gray studies are found significantly lower than those taken in the red, yellow, and light blue studies. And in fusobacteria values, we see that this study, as well as in the red study, they have significantly lower values than those in other studies. The reason this figure is important is because it shows us the comparison of components of the gut microbiome in different regions of the world that have been studied, how things have changed since their time in hospitalization, and the diets of the turtles. The authors mentioned that Firmicutes as common components found in the gut microbiome of many herbivorous reptiles. C. Caretta is known as a carnivorous species, which makes it surprising to find such a high abundance of Firmicutes and confirms that these turtles may also be feeding on seaweed, algae, as well as wood and debris. The authors also speculate that the differences seen in the Bacteroidetes and the Proteobacteria ratios from their data can be linked to a different diet, health conditions, or type of sample, as the yellow sample used cloacal swabs. And the author discusses how a high abundance of Proteobacteria can be a sign of dysbiosis, or it could represent a psychologically and metabolically assorted group that can be relevant for maintaining gut pH and for producing CO2 and nutrients for further colonization by strict anaerobes. This is seen in the blue study for green turtles, where the high abundance of Proteobacteria can either be seen in rehabilitation, which suggests their role in gut homeostasis. But the authors were also happy to see that they did not find Fusobacteria as a dominant phylum in the stool samples of C. Caretta, because they are commonly isolated from infected animals, but surmised that Fusobacteria abundance increases in sea turtles after many days of hospitalization. The authors noted as well how there was a comparable abundance of Bacteroidetes found in all microbes of sea turtles investigated so far, and explained how enzymatic and regulatory activities of Bacteroidetes may contribute to the digestion of accidentally ingested food containing carbohydrates in turtles, as many members of Bacteroidetes show an elaborate apparatus for acquiring and hydrolyzing otherwise indigestible dietary polysaccharides.
Okay, we also wanted to show this figure from the paper because we thought that it gave a good representation of the difference between what happens once you hospitalize a turtle and how that changes their gut microbiota. So it was a principal coordinate analysis, which I'm not going to go too in depth about because this isn't our most important figure, but basically the cluster of S samples on the left side of the graph are all samples that were collected 2 to 7 days after hospitalization, versus the 1S sample and the T samples that are clustered on the right side of the graph were taken from a range of 28 to 41 days after hospitalization, and then the S sample, S5, was taken 78 days after hospitalization. So this just shows how the gut microbiota changes over time when turtles are hospitalized. And as Joe said, the Fusobacteria abundance increases with hospitalization is the conclusion that they found from this graph. So then going over our conclusions and importance again, basically the gut microbiome similarities between turtles stranded on the Sicilian coast and other locations, they found that there were 3 core bacteria or families of bacteria that were found in all of these gut microbiomes, and it was the Firmicutes, Bacteroides, and Proteobacteria. And then metabolic requirements of microbial communities correlated with location and diet, as Joe was explaining. And also the differences in microbiota between rehabilitated and stranded turtles. So this is something that is important because it relates to the dysbiosis concept, which we talked about in class the other day, which is basically just a disruption of the microbiome within the turtles. So when they are rehabilitated in the hospital, this often leads to dysbiosis. And then also another conclusion is just the knowledge of gut microbiome and how that can aid in rehabilitation therapies. So in terms of future research, these are ones that were recommended from the paper in terms of just broadening their sample size, because they only took 9 samples, and also looking at more locations just to increase the understanding of gut microbiome and diversity within this population of loggerhead turtles. And then comparing gut microbiome to the environmental microbiome and seeing how these beaches that they are stranded on can have an impact on their gut microbiome, and also determining the function and associated benefits of each microbe that is found in the gut microbiota. And then some future research that we suggested as well that we came up with was sampling rehabilitated turtles, their gut microbiome after being reintroduced in the ocean, if they are ever reintroduced, and seeing how maybe dysbiosis has an effect on their survival once they're in their natural habitat again. And then also sampling gut microbiome of turtles in the ocean and comparing that to stranded turtles to see how being stranded itself affects their gut microbiome. And then another one we thought what might be interesting was maybe if looking if antibiotics could help combat the dysbiosis that is experienced in turtles when they are rehabilitated in hospitals. So yeah.
Save the turtles.
Save the turtles.
Thank you.
Thank you.